In the first three parts of week 3 of Advanced Immunology this week, we've begun by considering T cell activation. We've really focused this week on our CD4 positive T cells. We understand the role now of cytokines in T cell differentiation. We can see that there is a range of different CD4 positive T cells, going from Th1, Th2, Th17, and T regulatory cells. How the Th1, 2, and 17 are effector CD4 positive T cells. The T regulatory are suppressive or homeostatic T cells. Each of these different mature CD4 positive T cells is driven by the presence of certain cytokines, interleukin 12, interleukin 4, TGF-beta, interleukin 6, and in turn, the mature CD4 positive T cell produces specific cytokines which are directly related to the function of that cell. What we're going to look at here is in more detail the reciprocal regulation of T helper 1 and T helper 2. Our immune system relies on specific checks and balances. If one particular arm of our immune system is activated, the other arm of our immune system may suppress that or modify it. As we will see, if we have a strong Th1 response, it tends to dampen a Th2 response, and a strong Th2 response tends to dampen a strong Th1 response. This is all directly relevant to understanding allergy. So let's look first at a couple of terms. Atopy refers to the genetic tendency to develop allergic diseases such as allergic rhinitis, asthma, and atopic dermatitis, or eczema. Atopy is typically associated with heightened immune responses to common allergens, especially inhaled allergens and food allergens. So the question we're addressing here is, how does our immune system maintain homeostasis in terms of cytokine production and the number and activation of T helper subsets? In other words, why doesn't our immune system go out of control? Why don't we end up with too many Th1 or Th2 or Th17? Of course, chronic disease, that's exactly what happens. And that's one of the major challenges of immunology, is to start to unpack those changes which lead to this imbalance in the T helper subsets. So what we see in this rather complicated diagram is really showing us the interactions or the interrelationships between each of our different CD4 positive T cells. What we can see is we have our cytokines, interleukin 12, 18, and 27, for example. Interleukin 12, as we've already talked about, is critical for T helper 1 cells. Autocorine production and activity of interferon gamma drives T helper 1 cells. A similar role is played by IL-4 on our T helper 2, TGF-beta on our T regulatory T cells, and in our Th17s, we have TGF-beta plus interleukin 6. If we consider the relationship between T helper 1 and T helper 2, we can see that interferon gamma promotes Th1 but inhibits Th2. Interleukin 4 promotes Th2 but inhibits Th1. Th2 and T regulatory T cells, interleukin 4 will dampen T regulatory T cells, whereas TGF-beta will dampen T helper 2 responses. Tregs and Th17s is similar. TGF-beta by itself will dampen a Th17 response, but in the presence of IL-6, we will see a promotion of the Th17 response. So what we can see is that the activity, the activation of these different immune cells are controlled by cytokines which can stimulate one cell type while at the other inhibiting the activity of another T helper or CD4 positive T cell. So there are checks and balances in the production and activity of these CD4 positive T cells, which is all driven by cytokines. So what I hope you can now realize is that if we see a chronic inflammation where we start to see, for example, too many T helper 1 cells, which is a case in psoriasis, or too many T helper 2 cells, which is what we see in asthma, that this is probably reflected by increased amounts of specific cytokines. For example, in asthma, where we see lots of T helper 2 cells, you might reasonably assume that someone who has asthma also has lots of interleukin 4 being produced. And that is indeed exactly the case. Whereas someone with a very strong Th17 response has lots of interleukin 6. And again, this is exactly what we see. So what you might also be thinking is, well, if you have a particular illness, a chronic inflammation, by looking at cytokine profiles, by looking at T cell subsets which are defined by their cytokines, we might be able to understand the underlying nature of the disease. And that is exactly what immunologists do. So as I've said, T helper 1 and T helper 2 CD4 positive T cells undergo reciprocal regulation. And we see that this is through the cytokines interferon gamma and interleukin 4. So the cytokine environment is important for the differentiation of CD4 positive T cells, as well as for the activation and recruitment of cells of the innate immune system. If we think about CD4 positive T cell differentiation, what we see is that initially we have our naive T cell, and this is in the lymph node, of course. It sees antigen presented by the T cell receptor by the MHC2 in the case of our CD4 cells, and that this, with specific cytokines, leads to a pattern of cell differentiation. Cell differentiation is reflected in changing patterns of gene expression, the expression of cell-specific transcription factors, as well as specific cytokines. This differentiated T cell, therefore, is characterized by lineage-specific transcription factors, as well as cytokine production. So let's see what we have here. This is a rather complex-looking diagram, but let's just focus on one small part at a time. As you can see, what we're looking at first of all is the Th1 program. As we've heard before, it all begins with IL-12 in the company of the activated T cell receptor. And we can see that in these two green boxes. So we can see that IL-12 binds an IL-12 receptor, activates the transcription factor STAT4, and that this signal converges with our activated T cell receptor. This activity of interleukin 12 in turn activates the transcription factor TBET or TBX1 that we can see in here. This is a Th1 specific transcription factor. IL-12, TBET leads to the production and autocrine activity of interferon gamma, reinforcing the T helper 1 subtype. Interestingly, what also happens is that the interleukin 12 dependent production of TBX1 in the presence of interferon gamma now inhibits GATA3, which is a Th2 specific transcription factor. So if we have a naive T cell, it is of course undifferentiated, and it can differentiate into any one of the CD4 positive lineages. As soon as a cell starts to commit itself to one lineage, however, for example, Th1, it turns on all the genes which define that cell as Th1, and it turns off all of the genes that would otherwise have defined it as Th2, for example. And it does that through the repression of genes that express other transcription factors. Now let's look at T helper 2, and again, it's a similar pattern. So this is shown over here in blue, and as we know, the T helper 2 program begins with the cytokine interleukin 4. And again, in the presence of the activated T cell receptor. So now we're seeing T cell receptor, interleukin 4, the transcription factor STAT6, and the production of the cytokine, the Th2 specific transcription factor GATA3, and other T helper 2 cytokines. And that's indeed what we see here, where GATA3, which is a T helper 2 cytokine, can now also inhibit interleukin 12 as well as TBX21 or TBET. So we're seeing the reciprocal inhibition of T helper 1 gene expression programs in our T helper 2 cell. Finally, this leads to the reinforcement of interleukin 4 production and the autocrine activity of interleukin 4, further reinforcing the T helper 2 subset. So in summary then, what we're really seeing is that we have a naive T cell, and if you like, it has the potential, it's a type of stem cell, it has the potential to differentiate into a number of lineages. As soon as it sees a specific cytokine, however, interleukin 12, interferon gamma, or IL-4, it already starts to commit to a specific lineage. This commitment to a specific lineage opens up, drives, turns on genes for that lineage, for example, TBET, interleukin 12, interferon gamma in the case of Th1 cells, and turns off genes in other lineages. In the case of the Th1, that cell would turn off interleukin 4. It would turn off GARTAR3. And of course, GARTAR3 in turn will inhibit Th1 programs such as interleukin 12